All right, so we finally have an opportunity to integrate kind of all of the concepts that we've been talking about thus far, and that's when we get into this B cell activation, particularly the T helper dependent B cell activation. But before we do that, let's just take a step back here. So remember, when we're thinking of B cells, we're thinking a lot about humoral immunity. We're kind of looking at the end goal here of essentially generating antibodies and potentially a memory response, right? And so at the end of the day, when I'm thinking humoral immunity, I'm thinking of the end goal here is going to be producing antibodies, right? That's going to be immunoglobulins and particularly IgG in the setting of some memory response, okay? And the whole point of, immunoglobulins and antibodies, if you think about them, what are they doing? They're floating around usually freely in the bloodstream, right? And they're eventually going to attack or bind to extracellular pathogens. Okay, so that's going to be the key to humoral immunity. We're going to be very effective at targeting extracellular pathogens because we have all these antibodies floating around in the bloodstream. It's not going to be as effective against intracellular pathogens. Intracellular pathogens, I'd want something like cell-mediated immunity, where I can identify viruses or intracellular bacteria using T cells specifically, whereas with humoral immunity in general, this is going to be more effective against extracellular pathogens. Okay, so that's the concept here. So let's just erase this for a second and let's kind of talk about what we know so far so we can put this all together. So we're going to recap what we talked about in the T cell video, and we're going to go through it a little bit faster because we already talked about it with T cells. But essentially what we're talking about here is, right, we had some bacteria, for example. Okay, so we had some bacteria or some pathogen potentially, and then we had a dendritic cell around. Okay, so then we had some dendritic cells. So I'm just going to kind of draw it like this. This is my dendritic cell, and this dendritic cell phagocytosed this bacteria, okay? And then essentially presented components of that bacteria to a T cell, right? And so it's going to present these antigens. Usually because it's an antigen presenting cell, it will present them on MHC2. Presents this antigen from the bacteria on MHC2, and it's going to present that to a T cell. Okay, so then let's just say we have some T cell here. Right, so we have some T cell, and so here's my T cell, and this is going to be usually a naive T cell. It hasn't yet kind of ran into its pathogen, and on this T cell is going to have a receptor that's going to bind to that MHC2, and you might know that receptor by now. That receptor is going to be the T cell receptor, and it's going to co-bind with CD4. Right, so the T cell receptor, CD4, is going to bind to this MHC2, and this antigen is presented, and then there's going to be a co-stimulatory signal that we talked about in the T cell video, right? There's going to be a co-stimulatory signal, and that's going to be this CD28 binding to B7, okay? And this B7 is also sometimes written as CD8086. Okay, so this is all stuff that we talked about in the T cell video, and it's all recapped right here. We had a bacteria digested by an antigen-presenting cell, presented that on MHC2, usually in a draining lymph node, to a T cell receptor, CD4 positive co-stimulatory signal. Remember your rule of 8s, 4 times 2 is 8. That's how you know this is CD4. And again, it's an antigen-presenting cell, which usually was going to be MHC2. And then you have a co-stimulatory signal that helps this activation of the T cell occur, and that's going to be B7 and CD28. Remember, 7 times 4 is 28, right? So that's how we remember that. So that's essentially the T cell process. Once this happens, now this T cell can say, okay, is this thing you digested more of an intracellular bacteria like tuberculosis, where I'm going to release interleukin 12 and then cause a T helper 1 response? Or is this more of an extracellular bacteria that I want to generate more of a T helper 2 response? For example, if this was an extracellular bacteria, which is primarily what we're talking about with humoral immunity, right? That's what we just said. In that situation, we would want to generate a T helper 2 response. So if I want to generate a T helper 2 response from the previous videos, what do I want to release here? Well, remember, T helper 1 response would be interleukin 12. T helper 2 response would be interleukin 4. So this antigen presenting cell might say, hey, we need that humoral immunity, right? Because this is extracellular. I'm going to target this T cell with interleukin 4. Interleukin 4 will cause this T cell to go down the T helper 2 route. Okay, we'll go down the T helper 2 route, and we'll talk about the significance of that in just a second. So that's where we're at. Now, before we go any further, if this is kind of too sloppy for you, I have it all written here too. So you can see the antigen presenting cell presenting MHC2 to T cell receptor CD4, and then you can see CD8086, which is also known as B7, right? So this is also, let me write this in here, this is also B7. Binding to CD28, releasing cytokines, right? If it's interleukin 12, then it would stimulate the T helper 1 response. If it was interleukin 4, this would stimulate the T helper 2 response, right? So this is all the things that we just kind of talked about already. And that's where we're at. So we're activating a T cell. Now, the B cell, like I said, can also do the same things as an antigen presenting cell in the sense that it can also interact with bacteria and then present them on these immunoglobulin receptors. And that's essentially what you see here. So let's just say that we have a B cell here, and maybe there's some extracellular bacteria here. So this B cell might come into contact with this bacteria or potentially this bacterial antigen, and maybe it's a perfect fit for its B cell receptor up here. Okay, so I'm just going to draw it up here on the B cell receptor. Then what the B cell can do is it can internalize this antigen or bacteria and then represent it on MHC2, just like the antigen presenting cell did. So I'm going to draw an MHC2 here. And so we can put it on, let's just say this is our MHC2 receptor. Okay, so the B cell can place it now on this MHC2 receptor. Remember what we said, what's an MHC2 receptor? Essentially, it's allowing communication with T cells. Okay, so once this B cell binds to that antigen using its antibody, internalizes it, and then represents it on MHC2, now it can go to this step where the B cell can bind using that MHC2 receptor to a T cell. Okay, and that T cell can use its co-stimulatory signal. Again, it's going to be CD4 because we're talking MHC2, and that can help activate this B cell via a T helper dependent response. That's where this T helper dependent comes in. You have a T cell that is interacting with the B cell in order to stimulate the B cell to then go on to have B cell proliferation and eventually the formation of antibody secreting plasma cells. Remember, the end step of humoral immunity is essentially producing antibodies, and this T helper response is assisting with that. It's kind of giving it a huge upgrade in terms of giving the B cell the signals to proliferate, to undergo affinity maturation and isotype switching. Okay, so that's what's happening. This is very similar to what we saw with the antigen presenting cell. It's just when it happens with a B cell, it can then go on to form a humoral response. That's the difference. Okay, so that's essentially what's happening. Now, there's a couple kind of nuances to this that I haven't told you yet. So sometimes when you digest these antigens, these B cells, instead of them directly going to bind to T cells, sometimes these B cells can actually just become plasma cells themselves. Okay, so sometimes this B cell can just become a plasma cell instead of even going to the T cell. Now you might be saying, well, why doesn't it just do that? Why would I go through all of these steps to make a plasma cell when this B cell can essentially just do it on its own? What's the point of that? Well, when the B cell becomes a plasma cell strictly because it bound to its antigen and never interacted with a T cell, in those situations, that's a T helper independent response. In other words, the B cell never really interacted with a T cell. It's completely independent. The B cell is on its own. Okay, it didn't need the T helper cell to get activated. Now, the downside to that is typically when this happens, response that's generated is much weaker. Okay, it's much more weakly immunogenic, and that's sometimes how it's said. Now, to organize your thoughts here, you want to think about this in terms of proteins or peptides, right? Proteins, peptides, or you want to think of non-proteins or non-peptides. So things that are like carbohydrates, for example, like lipopolysaccharide, for example, would be a non-peptide. Lipopolysaccharide, LPS, on gram-negative bacteria, it's a polysaccharide, it's a carbohydrate. It's a non-peptide. Things that are non-peptides are not going to be able to be presented to T cells. T cells will only recognize peptide antigens. In other words, they'll only recognize protein-based antigens or peptide-based antigens. Okay, so if something's non-peptide and it's a conserved sequence, right? Something like LPS, something like flagellin. And we already talked about these conserved sequences. We gave them another name before though. Okay, when we first started this video series, we said the name for these are pathogen-associated molecular patterns, right? Or PAMPs. And these are what are recognized by toll-like receptors or pattern recognition receptors, right? To stimulate that initial immune response that goes on to generate interleukin 1, interleukin 6, TNF alpha, right? So these are these same structures. And so you might be saying, okay, well, they set off the immune response. Why do we have to have this T-independent response? The reason is because the T-independent response is what allows us to generate antibodies like IgM, for example. These plasma cells that are activated independent of T cells because there was a carbohydrate antigen, for example, bound here, they will go on to release IgM, which is kind of like your standard immunoglobulin. Now, it's important to note that IgM is not really involved in the memory response. It's kind of like the initial response to an infection from the immune cells, right? You have your inflammatory response from neutrophils, which is largely driven by pattern recognition receptors and toll-like receptors recognizing LPS and flagellin. The immunological response, right, from B cells is driven largely by this T helper independent response, where B cells can bind to LPS, can bind to carbohydrates, right? But instead of going to the T cells, they just become plasma cells and release IgM. What I'm saying here is this is more or less driven by the T helper independent response. That's why it's all in orange here. Whereas if you go up this way, right? If you go up this way, this is more so the T helper dependent response. And a lot of this is driven by the type of antigen. Is it non-peptide like flagellin or LPS? Our pathogen-associated molecular patterns are very classically going to fit into this category, things that are non-peptide based, and not all of them necessarily fit into that category, but that's one way to think about it. Or is it going to be a peptide antigen? If it's a peptide antigen, it's most likely going to go down this route. Now, some peptide antigens will also kind of just generate an IgM response, okay? But more or less, the way that you want to think about it is the way that we have it kind of listed here. Now, let's just clean this up for a second. And so with the T helper independent response, right, these specific carbohydrates cause cross-linking of the surface antibodies. That's how this B cell will get activated by non-peptide antigens, and that will lead to production of IgM, which is what we just said. The T helper dependent response involves T cell activation via an antigen presenting cell. That naive B cell is going to bind its antigen to the B cell receptor. That's what we just said here. So antigen binds to the B cell receptor. That's the step. Then the B cell receptor has mediated endocytosis of the antigen. That's the step, right? It internalizes that antigen. Then the next step would be to display that antigen on the MHC2 receptor, which we can see here. So we found the antigen bound to our immunoglobulin, we internalized it, and then we presented an MHC2, and there you can see it right there, bound to MHC2, binding to a CD4 receptor, right? Activated T helper cell binds to that B cell using a T cell receptor and CD4 positive co-receptor. And then the last step here, there's another co-stimulatory signal. And this signal, CD40 ligand co-signal, essentially involves CD40 ligand on the T helper cell and CD40 on the B cell. And this specific step is involved in isotype switching. And that's very high yield to remember that CD40 ligand, CD40 binding, and this is one of them that I would absolutely remember, is involved in isotype switching. So it essentially will cause this antibody to change its heavy chain. Okay, so remember, you have a variable region out here that binds to a very specific antigen, and then you also have another region down here that's the heavy chain. So the heavy chain will tell you, are you dealing with IgE, IgA, IgM, but the actual variable chain is what binds to that specific component of a pathogen. Now, once this B cell goes through this whole process, presents on MHC2 to the T cell, and CD40 ligand interaction occurs, you're going to have specific interleukins that can be released. Okay, and we already talked about this a little bit when we talked about the cytokines. These different interleukins can cause different types of responses. So for example, if this T helper cell releases interleukin 4, that will stimulate mostly IgE, but also some IgG response. Interleukin 5 is going to be IgA. Interleukin 13 is also IgE. So just make sure you're familiar with these, particularly interleukin 4 and interleukin 5, probably being the two big ones I would remember. Once that happens, those B cells can then go on to proliferate. Okay, so the B cells will become activated, they'll proliferate, and then they can eventually become plasma cells, which are essentially going to be antibody factories. And if they, were stimulated with interleukin 4, they might be pumping out a bunch of IgE or a bunch of IgG, for example. Some of these B cells, through this proliferation, will eventually become memory cells, so that if we ever interact with that antigen again, we'll have a much more robust response in the future. Okay, so that's where the memory response comes from. Now note, in the T helper independent response, right, when we went down this road over here towards this IgM, we never generated memory B cells. We only generated plasma cells. Okay, so that's why there's not a really strong memory response when I have T helper independent antigens. Okay, I need T helper dependent antigens to generate a very robust memory response. This is where it all comes from, is from this pathway. Now, the last thing on here is this affinity maturation. So what is that talking about? So affinity maturation is essentially talking about the B cells that have these kind of variable regions up here that bind to specific antigens. What we want to do is we want to make that binding as strong as possible. And so once this interaction occurs with the T cell and the B cell, at some point these B cells will eventually migrate to a lymph node and, like we said, go into the secondary follicles, and you'll have your germinal centers that are going to have proliferating B cells in there. Inside of those proliferating B cells, there's going to be very slight variations that are going to happen to this variable region. And the idea is we know that there's some antigen that is able to bind to that B cell receptor. But what we want to do is we want to maximize that binding. So we kind of know we have the right formula. We just want to make little tweaks to it, and we make these little tweaks to a whole bunch of different B cells. And then the B cells that bind the strongest, right, from those tweaks are going to stick around. And so there's random mutations of the B cell receptors so that only the strongest affinity B cells survive. And again, this is all happening in the lymph node, in those germinal centers that we talked about in the last few videos. And again, here's a lymph node, right? It's going to be more of those pale staining regions in the lymph node where you have the germinal centers. And again, we're just trying to keep the strongest binders in there. And essentially that's what affinity maturation is. Again, we know we have something that binds, we're just tweaking it to get the absolute strongest binding. And again, you get affinity maturation largely driven by T helper dependent responses. So both affinity maturation and isotype switching are a result of a T helper dependent response and not a result of a T helper independent response. That's particularly important. So again, we got isotype switching because of the T helper interaction. We also get affinity maturation because of the T helper cell interaction with the B cell. If I don't have this interaction, if I just go this way, I'm just generating IgM kind of blindly here against that antigen without developing a robust immunogenic response. That's the key. So just keep that in mind. And you might be saying, well, is this applicable at all in real life? I mean, it absolutely is. I mean, if you think about things like vaccines, for example, right? So, and we'll talk about this when we talk about vaccines here, but, your strep pneumo vaccine, right? There's different variants of it. The PPSV23 variant is going to have a capsular polysaccharide. So the PPSV23 vaccine for strep pneumo is going to have a capsular polysaccharide. And this is what generates the immune response. Okay, so it's going to be the polysaccharide capsule. Remember what we're talking about. It says it right there. Polysaccharide. What's a polysaccharide? It's carbohydrate, right? Go back to your biochem. So this is going to be a carbohydrate. A carbohydrate, is that a peptide? No, it's not a peptide. And if it's not a peptide, then it must be a T helper independent antigen. Okay, so if I give somebody this vaccine, I'm giving them a T helper independent antigen, right? It's an independent antigen. So I know that I'm not going to get this interaction with the T cell. I'm going to go down this road. So do you think I'm going to get a strong immunogenic response if I give somebody a vaccine that has a T helper independent antigen or a polysaccharide? Probably not. I'm not going to get a really robust immune response. That's why we add things like adjuvants to these vaccines, for example, or that's why a peptide vaccine in some situations might be stronger. And in a lot of these situations too, if you're giving these independent antigens, you'll see there's in some situations there might be more boosters involved potentially. But I don't want to get too off track. That's the general concept. And if you got to this point in the video series, kind of building up the material, adding up the layers, this is kind of like the peak. And from this point forward, we can start integrating all of these basic science concepts to understand the pathology and some of the implications.